Good morning, class, or good afternoon, or good day. Today we are going to review meiosis and genetic variation. So, meiosis and genetic variation. So, the key concept of your notes today is you will learn that during meiosis, diploid cells undergo two cell divisions that result in haploid cells. So, once again, diploid cells undergo two cell divisions that result in haploid cells. So we're going from two sets of chromosomes down to one set of chromosomes. So cells go through two rounds of division in meiosis. The purpose of meiosis is to reduce the chromosome number and to create genetic diversity. So reduce the chromosome number and create genetic diversity. As we see here in meiosis, remember as we remember, meiosis is the process of making sex cells or gametes. So meiosis produces genetically unique cells. It results in haploid cells. It takes place only at certain times in an organism's life, and it's involved in sexual reproduction. Meiosis 1 and meiosis 2 each have four phases that are similar to those in mitosis. Pairs of homologous chromosomes separate in meiosis 1. Homologous chromosomes are similar, but they're not identical. And as you recall in the last lecture, we talked about how you get one pair of homologous chromosomes from each parent. At the end, the sister chromatids divide in meiosis 2. Sister chromatids divide in meiosis 2. And the sister chromatids are copies of the same chromosome. So they're copies of the same chromosome, as we see here. So if I look on the illustration, here are the homologous chromosomes. So the whole X would be considered a homologous pair, okay? Each individual half of a chromosome is known as a sister chromatid, Meiosis 1 occurs after the DNA has been replicated. And what meiosis 1 does is it divides the homologous chromosomes into four phases, as we see here: prophase, metaphase, anaphase, and telophase. Okay, so if we look at humans, we start off, right, with our germ cells. Okay, so we have a total of 46 chromosomes. Now, just like meiosis has an interphase. In an interphase, the DNA has to be replicated. So if I replicate the DNA, meaning that I make a copy of my 46 chromosomes, so 46 times 2 is going to give me, let's see if you guys have your mental math, 92 chromosomes. Okay, 92 chromosomes. So at the start of meiosis, we have a total of 92. Why? Because like we said, the DNA has been replicated. So as it's replicated, it makes obviously a copy of itself. Then in meiosis 1, the homologous chromosomes are going to separate. Before they separate, they're going to do something unique. And what they are going to do is they are going to line up in the middle, separate, and then pull apart. However, during meiosis 1, pairs of our DNAs are going to switch information, which is a process known as crossing over. And we'll get to that in a minute, but this process is what actually makes each cell genetically unique. So meiosis 2 divides the sister chromatids in four phases. So DNA is not replicated between meiosis 1 and meiosis 2. So the DNA is no longer replicated. It only replicates during meiosis 1. So as we see here, in meiosis 2, we go through the same phases: prophase, metaphase, anaphase, and telophase. Notice the difference between picture number 5 and picture number 8. So as I see 5 and 8, okay, So we have our chromosomes and then we have our chromatids. So we also make 4 cells at the end. Each one of these, n equals 1, meaning that each of these sex cells has one set of chromosomes, one set, okay? They are all haploid cells. They're all haploid cells. All right, so just to go over meiosis. So in the beginning, at the start of making our sex cells during interphase, here's our germ cell. So remember, this is the master cell. This is either found in the testes or the ovary, okay? Humans, as we said, have a total of 46 chromosomes, okay? During interphase, the chromosomes duplicate. So they make a copy of themselves. So we go from 46 to 92. Okay. Then it's going to go ahead and go through the prophase, metaphase, anaphase, and telophase. So we have prophase 1. Notice how the chromosomes are visible. Now, in this illustration, I also want you guys to recognize how these chromosomes here, the red and the blue, they are starting to cross over, meaning that their DNA segments are switching places or they're shuffling. This information here, the shuffling process, accounts for genetic variation. Okay. Once these chromosomes have exchanged DNA, then they're going to randomly line up in the middle during the process of metaphase. Anaphase, they separate. And telophase, we have 2 new cells. Now notice here, see how the color change? We go from red-blue separated to red and blue integrated, okay? At the end of meiosis, we have 2 cells, right? So now 92 has been separated in half. So we should have 46 here and 46 here. Now we're not done, okay? Why? Because we need to reduce the number down to 23. So it's going to go ahead and begin at prophase 2, metaphase, anaphase, and then finally telophase, where we actually make the sex cells. So at the end, so same thing, chromosomes are visible, they line up in the middle, they start to pull apart, and at the end, we have 4 cells, each with 23 chromosomes. Now, each of these are different. They have genetically different information, and you can visually see the chromosomes here, how the chromosome color makes them look different. Why? Due to crossing over that has occurred in prophase 1. Crossing over, and then the chromosomes randomly lining up in metaphase 1. So basically, it's like I shuffle the cards and then I deal them out. So when I shuffle, I change the genetic information, and when I deal them, it's another change of the genetic information. Right. Meiosis differs from mitosis in significant ways. So meiosis, as we said, has 2 cell divisions, while mitosis has 1. So meiosis has 2 cell division, mitosis has 1. In mitosis, the homologous chromosomes never pair up. So in mitosis, homologous chromosomes never pair up. Meiosis results in haploid cells and mitosis results in diploid cells. So remember, in meiosis, we're making sex cells that has half the number of chromosomes. And in mitosis, you're making body cells. In body cells, you retain the chromosome number. Right? Eventually, our haploid cells are going to develop into mature gametes. So they have to develop into mature gametes. The gametogenesis, that's a term you should know, is the production of gametes, or it's the term used to make sex cells. Gametogenesis differs between females and males. So it differs between females and males. In males, the sperm becomes streamlined and motile. Motile meaning that they grow a tail, they're able to move. And sperm contribute primarily DNA to the embryo. So when the egg and the sperm unite during the process of fertilization, The tail is going to come off, and the only thing that the sperm is going to contribute is the DNA. So the nucleus from the sperm and the nucleus from the egg cell are going to merge together, and then the DNA or the homologous chromosomes are going to look for their matching pair, and then they're going to form the embryo. For females, eggs contribute DNA. Eggs contribute DNA, cytoplasm, and the organelles. So all your organelles come from your mother. They come from your mother. During meiosis, the egg gets most of the contents and the other cells form polar bodies. Now notice the difference between sperm production and egg production. For every one germ cell, a male will produce 4 sperm cells. Okay? Now notice for the egg cell, for every one germ cell, a female is only going to release 1 egg. The others become what are known as polar bodies, okay, or immature eggs. So I want you to think, why is it significant or why is it important for a female to release only 1 egg and not 4 eggs? Why is it that we women do not release 4 eggs at each cycle? Why does she only release 1? Take a minute to think about that. Why does the female only release 1 egg? I'm hoping you said because if she released any more than 1 egg, she will have multiple children. Okay. So if an egg and a sperm unite, they form an embryo, right? So there's 2 eggs, there's 2 embryos. 3 eggs would be triplets and 4 will be quadruplets. Okay. So women's bodies are usually only adapted to carry 1 child at a time. 2 once in a while, right? Which is rare. But any more than 2 will put stress on the woman's body. And for that, she'd probably have to be in medical care to make sure that everything is okay with the babies, right? So that is a little FYI. All right. I want you guys to go ahead and watch this video. I'm not going to play it during my lecture, but please watch the difference between identical versus fraternal twins. Identical versus fraternal twins. All right, here's our illustration between the difference of mitosis versus meiosis. So remember, in mitosis, you're making body cells or somatic cells, which retain the same number of chromosomes, meaning we're making identical cells. And in meiosis, we're making sex cells. Each would have genetically unique information. And from that cell, we make four cells. Once again, here's another video on the comparisons between mitosis and meiosis. Please make sure to watch this as well. All right, in our next section, we're going to talk about genetic variation and the importance. So the key concept is that independent assortment and crossing over during meiosis results in genetic diversity. So go ahead and take a look at these illustrations. So we see the dog, we see these clamshells. Notice how in dogs, particularly, right, there's a lot of genetic diversity within dogs. Well, why is that? Okay, dog is a dog. They're the same species, but they do get genetically different alleles. They get genetically different alleles and traits, okay, which make them look different. Same thing with our oysters, or I'm sorry, our clams. Okay, so sexual reproduction creates unique combination of genes, right? Go Dodgers. So as we see in this illustration, there's a big picture of humans, and notice that no 2 humans in this picture are identical. They are each genetically unique from each other. Sexual reproduction creates unique combination of genes through 2 processes. I'm sorry, 3. Independent assortment of chromosomes in meiosis, crossing over, which isn't stated here, and then the random fertilization of gametes. So unique phenotypes may give a reproductive advantage to some organisms, So an example of a phenotype would be like the ability to camouflage, right? So some animals are able to camouflage. If you can't camouflage and you stick out, chances are that predators will see you and you'd probably be eaten, okay? That wouldn't give you a reproductive advantage. All right, crossing over during meiosis increases genetic diversity. So crossing over during meiosis increases genetic diversity. You need to know this. So crossing over is the exchange of chromosome segments between homologous chromosomes. It occurs during prophase 1 of meiosis 1, and it results in the new combination of genes. All right, so as we see in this illustration, if I look at picture number 1, the two homologous chromosomes pair up during prophase 1, right? Remember we said the DNA made a copy of itself. Now what's going to happen is in this position, some of the chromatids are very close to each other and their segments cross. Now some of these segments break off and reattach to the homologous chromosomes. So basically what's going on is, remember, you are made of 23 pairs of chromosomes. You get one set from parent 1, and then you get one set from parent 2. Okay. What's basically going to happen is your mom's DNA and then your dad's DNA is going to shuffle and exchange information, so that now when you pass on your DNA to your offspring, it is now a combination of your mom and your dad's DNA together. Okay. It's not separated, it's combined together. Okay. So as we see here, and this genetic reshuffling accounts for genetic variation. That's what makes us all look different from each other, especially like siblings. All right, here are some famous siblings. So if we look at genetic diversity and similarities, we can look at differences and similarities. So we have Beyonce, we have Lenny Kravitz and his daughter Zoe. We have Cardi B and her sister, the Wilson brothers, Hadid siblings, similarities and differences, right? The Gyllenhaals, so Jake and Maggie, they're siblings. The Jonas brothers, some of you guys already know, similarities and differences. And then obviously the most probably famous, which most people recognize, would be the Kardashians / Jenners. So similarities and differences. Okay. So remember, all of these siblings have some qualities where they look alike, some that they look different. And that's due to, as we said, crossing over and genetic recombinations. Now, chromosomes contain many genes. So if I were to think of a chromosome, like we said, it's a long strand of DNA. The farther apart two genes are located on a chromosome, the more likely they are to be separated by crossing over. Okay, so if I look at gene A, B, C, D, okay, notice how A and B are together and C and D are together. When they cross over, meaning when these segments switch information, genes that are really close together tend to be inherited together, which is called genetic linkage. So genetic linkage allows the distance between two genes to be calculated. So we can see how close or how far apart genes are based on these linked genes, Okay. An example of linked genes would be red hair and freckles. So the majority of people that have red hair usually have freckles. Why is that? Because those genes are so close together that they inherit together. All right, if there's anything, please make sure that you listen to this. What's the value of sexual reproduction? Okay, sexual reproduction introduces genetic variation. Okay, genetic recombination occurs in 3 ways: independent assortment, crossing over, and random fertilization. So independent assortment of chromosomes is when the random alignment of the homologous chromosomes in metaphase 1. Crossing over is the mixing of alleles. And random fertilization is which sperm fertilizes which egg. That's all done randomly. Okay? It's the driving force for evolution, and it provides variation for natural selection. Okay, provides variation for natural selection. All right, finally, how do we make sperm and eggs? You must reduce the number of 46 chromosomes to 23. 46 to 23. you must reduce the number of chromosomes by half, right? So notice here we have egg and sperm. These are our germ cells, okay? Through the process of meiosis, we go from 46 down to 23. So 46 to 23. Egg and sperm unite through the process of fertilization. So through the process of fertilization, so 23 and 23 are now going to form 46, which is going to be a zygote. Okay, a zygote is a scientific term for a fertilized egg. So that's when the egg and the sperm, their nucleus unite and the DNA merge together to form the zygote. Now, when the zygote starts to grow and develop, it's going to grow and develop through the process of mitosis. So now, through cell division, mitosis, these cells are going to grow into something that looks like a human. Okay. All right, finally, here's the last video on meiosis. So please go ahead and take a look at this video. It is with the Amoeba sisters and kind of summarizes everything that we talked about today. All right, guys, I hope you have a great day.